This is Rachel Saunders, and I'll be discussing how the polyglutamine repeats in Huntington's disease causes early mitochondrial calcium defects. In order to understand the relevance of calcium in Huntington's disease, you really need to first understand the role of calcium in mitochondria. Mitochondria are really important in making our cells function. They produce ATP, and that allows our cells to carry out their functions. Proper mitochondrial activity is critical to neural activity. As you can see in the image on the right, in average mitochondria, there is a standard uptake and retention of calcium. In the Huntington-affected mitochondria, there's a lower uptake and retention of calcium. Excess of calcium results in cell death. Obviously, this image is very simplified, but as you can see, patients with Huntington's disease have a lower calcium retention capacity than those without. The purpose of this experiment was to find the mechanisms, the ways in which the Huntington protein was causing this neurodegeneration. The researchers performed an experiment on two groups of mice. They were yeast artificial chromosome, or YAC, transgenic mice. One group had normal Huntington's protein, they called them YAC18, and the second group had mutant Huntington protein, which were called YAC72. They looked at the brain mitochondria of these two mice groups, and what they saw was that the mutant, YAC72, had lower calcium retention capacities. What does this mean for the mitochondria? Since the YAC72 mice had lower calcium retention capacities, the aggregated Huntington protein could associate with the mitochondrial membranes. Figure 4 in the paper is an image of brain mitochondria in these mice, both the YAC72 and 18. So on the bottom you can see a fraction of the image that we'll be discussing in the following slide, a little bit more in depth. But for now, you can just see this example. So what they did is they used a method involving immunogold particles. These particles bind to an EM48 antibody, which binds to the N-terminal mutant Huntington protein. The immunogold proteins can be viewed using an electron microscope. So on the left, as an example, you can see that black clump, and that's a cluster of Huntington protein. So in this slide, you can see the full figure, figure 4. Now on the top row, you can see the YAC72 mouse brain mitochondria, and on the bottom row, you can see the YAC18 mouse brain mitochondria. In the first row, as you can see, there are quite a few clusters of the protein, and they're right at the mitochondrial membrane. This revealed that because of their decreased membrane potential, the N-terminal mutants can associate with the membrane, and as a result, the aggregated proteins can associate with the membrane. What the authors ended up concluding was that the Huntington's disease is associated with the defectant mitochondrial membrane potential. At least part of the dysfunction in this disease can result from a direct interaction of the mutant Huntington with mitochondria. This paper shows the connection between Huntington's disease and mitochondrial dysfunction. And they used polyglutamine repeats as a means of finding the mitochondrial dysfunction, but they didn't take on the question of how the mutation happens originally. This paper would lend itself more to research for a cure than to research for prevention.